Hi, my name is Andrew Moraga, and I'm a third-year environmental science student here at Carleton University. Today, I'm going to be talking to you about my third-year project. I worked on it with my peers, Alexon and Haley. We were looking at the decline of honeybees and what's causing it. I'm going to be focusing on my section, which looked at pathogens. First off, let's talk about honeybees. Honeybees, specifically what we looked at, are Apis mellifera, the western honeybee, and they're a commercially important species to us. Why, might you ask? Well, they're responsible for over $200 billion worth of pollination services worldwide. As you can see in my image, these boxes, they're honeybee colonies. Inside each box there are frames, and these frames contain the honeycombs which the honeybees use to store their honey. Humans maintain these colonies for our commercial purposes for agriculture. The decline of these honeybees is what's the problem and what is our concern. In 2012 to 2013, nearly 50% of commercial A. mellifera colonies were lost in the US. The death of these honeybees has been characterized now as colony collapse disorder. What is colony collapse disorder? Well, it's characterized by a sudden disappearance of the worker bees. This is important because honeybees are eusocial insects. What does that mean? It means that they have overlapping generations living together, they have a cooperative brood care, and there's a division of labor based on non-reproductive and reproductive roles. As you know, honeybee colonies have queen bees, and those are the ones that reproduce. They spread their genes throughout the colony, and all the worker bees are sterile female worker bees. They don't lay any eggs. Worker bees are divided into in-house and forager honeybees. The in-house bees are the ones from around 0 to 21 days old, and after 21 days, these in-house bees generally transition into a forager honeybee. Foraging is more risky than in-house tasks because it exposes them to factors outside of the colony. So why are they declining, you might ask? Pathogens. Well, at least that's what I looked at. There were two other areas that we looked at, which was the natural environment and chemical exposure. But today, pathogens is what I got, so pathogens is what I'm talking about. Immune health. The honeybees have a minimal amount of immunity genes, and that already puts their immune system at risk and makes them vulnerable to other factors and stressors. They have a limited amount of NF-κB transcription factors, which is a family of transcription factors. The first issue, or problem you might say, comes in with Varroa destructor, which sounds like a scary name, but it's just a mite, but a problematic mite. This mite attaches itself to the honeybee. As you can see from the image, it's a red sheet that covers the honeybee, and often there can be several of them on the body of a honeybee. They feed on the hemolymph, which is a liquid blood-like substance that flows through the honeybee. These puncture wounds cause the immune system of the honeybee to react, and since they have a limited amount of transcription factors, those transcription factors are concerned with the injury that this mite has caused. The mite often acts as a vector for a virus known as DWV. DWV is deformed wing virus, and as you can tell from the name, it leads to deformed wings in these honeybees. Honeybees need to fly to live. So if they have deformed wings, they can't fly. If they can't fly, they die. Ultimately, the lifetime of a honeybee with DWV is around 24 to 36 hours. Finally, we come to Nosema. Nosema is a fungus, and it has two strains of major concern, Nosema apis and Nosema cerana. These fungi infect the midgut physiology of the honeybee. This becomes a problem because this infection leads to increased energy demands. Increased energy demands leads to an increase in foraging tasks and behavior to deal with this increased energetic demand. This ultimately exposes the honeybee to more risk, leading to its death. If not led to death from foraging activities, the infection from the nosema will eventually lead to the death of the honeybee, blocking its midgut with all of its spores. What does all of this mean? Well, the varroa mite on its own usually will not kill the honeybees. However, the varroa mite with DWV or the varroa mite with nosema, or maybe even all three, is what ultimately leads to the death of the honeybees. Because of this compromised immune function, each single factor that builds up puts a greater and greater strain on the immune system of the honeybee. This makes it a problem because the resources they have available to deal with the stress are becoming thinner and thinner and thinner. So, what we have for mitigation efforts now, at least what research has come up with, is selective breeding of the queen. The queen is the one responsible for spreading genetic material throughout the colony. Therefore, whatever genes she has will end up being in the colony. If we increase her cleaning behavior or hygienic activities, then it's likely the worker bees will also continue this, being sure to keep these mites out of the colony and reduce or prevent the introduction of nosema into it. This would help control these issues inside of the hive and prevent them from becoming a major problem and ultimately leading to colony collapse disorder. Another suggestion is the reduction of harmful chemicals. Although they are not lethal to the honeybee, like I was mentioning earlier, they compromise immune function, and this compromise of immune function leads to vulnerability, which can ultimately lead to death. There's still a lot of research being done around this issue, and hopefully they come up with a way to prevent our honeybees from dying. Thank you for watching.